on the top. Check the body parts and make sure everything intact. Okay, check the body parts and make sure everything intact right now. Hey, your foot walking, your waist walking, your neck walking.
So today we'll be discussing the ATP generation in sprinters compared to distance runners. ATP is adenosine triphosphate. It is the currency of biological life, and it is the energy stored within a phosphate bond. ATP can be seen here, where this is the adenosine part, and these are the three phosphate groups. Now, to release this energy within the bond, it undergoes a hydrolysis reaction from ATP to ADP. ADP is adenosine diphosphate, and it also releases a phosphate group and energy. And the energy is released is used to fuel metabolic reactions. Now, this hydrolysis is a splitting of the compound into two smaller molecules, and it uses a water molecule. This reaction is catalyzed by adenosine triphosphatase, or ATPase. Now, there are three biochemical systems in the body that produces ATP: the immediate system, the short-term system, and the long-term system. The immediate system is fueled by ATP stores and phosphocreatine, and it occurs immediately starting exercise. This only lasts up to 10 seconds. The creatine is used when the ATP stores are depleted. The short-term system is the anaerobic part, where it follows the anaerobic fate of pyruvate from glycolysis, and it's fueled by glycogen, glucose, and glycerol. The glycerol can be obtained from triglycerides. It lasts up to 90 seconds of intensive exercise. The long-term system follows the aerobic fate of pyruvate from glycolysis and oxidative phosphorylation of certain compounds. It's fueled by the breakdown of carbohydrates, fatty acids, and amino acids. It can last up to a few hours. In fact, the long-term system, it's what normally occurs in our body. As you can see, they are muscular. Now, sprints are usually short distances, less than a quarter mile, and it's a short period of time, but it's intensive exercise. It's based on an athlete's strength and speed, and the immediate and short-term energy systems are predominant. Now, in immediate system, when ATP is depleted, the phosphocreatine is then used to generate ATP, and it's an organic compound found in muscle fibers and the brain. In the muscle fibers, it's used to power contractions, while in the brain, it's used to power neurons. Phosphocreatine is the phosphorylated form of creatine, and creatine can be synthesized by the liver from the amino acids arginine, glycine, and methionine, or it can be obtained from the diet, mainly from meat in the diet. It generates ATP when it's dephosphorylated, and the reaction is a reversible reaction. This occurs in the cytoplasm of muscle fibers, and the enzyme responsible is creatine phosphokinase. Now, glycolysis is the process by which glucose is converted to pyruvate, and glucose can be derived from glycogen, or it can be derived from a diet. Glycolysis occurs in a cytosol of cells. When glucose is obtained from glycogen, it's a process called glycogenolysis, which is the breakdown of glycogen. Glycogen is a polymer of glucose, and it contains alpha 1,4 linkages and alpha 1,6 linkages. In a branch, glycogen phosphorylase acts first to detach monomers until it has 4 monomers remaining. The glucotransferase then comes and takes the terminal 3 carbons and transfer it to the longer chain, leaving the glucose monomer with the alpha 1,6 linkage. The glucosidase is an enzyme, another debranching enzyme, and it breaks the alpha 1,6 bond to DC-glucose. When this is completed, it results in a straight chain, at least for that part, and glycogen phosphorylase can continue the process. Now, to regulate glycogenolysis, there are two hormones, glucagon and insulin. Glucagon results in glycogen being degraded, while insulin results in glycogen being synthesized. Glucagon results in a series of reactions and results in glycogen phosphorylase kinase being active, and this leads for glycogen phosphorylase to be in the active form, which is the A form, while insulin does the opposite. It stimulates protein phosphatase 1, and this inactivates glycogen phosphorylase to the B state and also inactivates glycogen phosphorylase kinase. Now, in glycolysis is a series of reactions to form pyruvate, and for every 1 molecule of glucose, you get 2 molecules of pyruvate. Now, as you can see, there are 3 irreversible reactions. There are also reactions that use ATP and that release ATP, and as you can see, NADH is also formed. So this is the investment stage where 2 ATP is used, while this is the payoff where 4 ATP is produced. So a net of 2 ATP is produced from these reactions. The main regulatory enzyme of glycolysis is PFK1, which is phosphofructokinase 1, and this glycolysis occurs in the cytoplasm or the cytosol of cells. Now, the anaerobic fate of pyruvate is where pyruvate is converted to lactate by lactate dehydrogenase, and this is because oxygen is not readily available for it to go on to the aerobic fate. When lactate builds up, it builds up that lactic acid, and it can cause cramps, discomfort, fatigue, and leave a person gasping for air. However, when oxygen is available again, it can be degraded back to pyruvate for it to continue on. Now, lactic acid is an acid, of course, and it results in a pH of the environment to be below the normal pH. As you can see, they are tall and skinny. Now, distance running is usually large distances, more than 5 kilometers, and it's a long period of time. It's based on an athlete's endurance and stamina, and the long-term energy system is predominant. Now, fatty acids are broken down in this system, and this occurs in the mitochondrial matrix. And the fatty acids, if they are long chain, they are transferred by the carnitine shuttle. If they are very long chain, they first go to the perioxysomes for preliminary breakdown, and then they come to the mitochondrial matrix. The short and medium chain, they don't have to go by the carnitine shuttle, they can be transported inside. Now, this is a four-step reaction. The first step is a oxidation reaction, or FADH is produced. The second is a hydration reaction, where water is incorporated. The third is another oxidation reaction, where NADH is produced. And finally is a dialysis. Now this is a cycle because acetyl-CoA is obtained, which is a 2-carbon compound, and the fatty acid is a long chain compound. So it continues this process until the maximum number of acetyl-CoA can be obtained. If there is an odd number, however, a propanol is obtained, and this has to undergo a 3-step pathway to break down. Now, to obtain fatty acids, we have to break down lipids, and also we can hydrolyze triglycerides into glycerol and free fatty acids. It's regulated by hormones, and the glycerol is ultimately converted to glyceraldehyde 3-phosphate, which can join glycolysis. Now, for the aerobic fate of pyruvate, the pyruvate is combined with CoA to form acetyl-CoA, and this acetyl-CoA joins the citric acid cycle, or the TCA cycle, or the Krebs cycle. They are all the same cycle. And from the cycle, you can see NADH is produced, ATP is produced, and FADH2 is also produced. Water is also incorporated. For amino acids, they are degraded into compounds that can enter the various stages. So they can be converted to pyruvate. They can also be converted to certain compounds in the TCA cycle. And do you remember the NADH and the FADH being produced? Well, they enter the electron transport chain, which is a series of electron carriers, and they result in ATP being produced. This is actually where most of the ATP is produced. These molecules, how they are electron carriers, H plus is basically considered, an electron. H plus is pumped in the mitochondrial space between the membranes, and this builds up. It results in a gradient, and it also builds up potential energy. And this potential energy is released when ATP synthase is open, and so there is a flood of H plus ions coming through this channel protein, and it converts the potential energy to kinetic energy, and this kinetic energy allows for ATP to be synthesized. Now this is just a table comparing sprint and distance running, and what we didn't mention before was the muscle fibers. The muscle fibers in sprinters are usually white due to the lactic acid buildup and the absence of oxygen, and in distance runners is red and is oxidative because oxygen is is used, and it's used to carry about the aerobic respiration. Now, this is just a chart or a graph, just comparing the speed and time using various data from the Olympic Games. And as you can see, as distance increases, speed decreases. It actually almost is halved. Exertion can be defined as the activity on your muscles in various forms to maintain fitness. Overexertion, however, occurs when the physical exercise goes beyond your body limits. It can result in nausea, vomiting, and fainting. It's associated with hypoglycemia, heat exhaustion, and dehydration. Hypoglycemia is due to low blood sugar levels. The normal levels are between 80 and 100. Heat exhaustion and dehydration can be the result of the water being used up, and if you are not replenishing the fluids in your body, the body won't be able to cool down your body by sweating because it doesn't have the water molecules to expand. And dehydration, it body lacks the water. So thanks for watching. I hope it was informative. So thank you again.